Thanks to our audience joining GU Oncology now, and thanks to David Braun. I am so excited to just be here and chat with you today. Congratulations, on your Nature paper and this huge undertaking. So many co-authors, so many amazing, science and correlates. And so just wanted to chat with you today and learn a little bit more about, what it took to make a trial like this happen, and what you think some of the main points were that you learned from this and kind of where you see it going next.
No, thank you so much for chatting with me today. It's something that it took many years. We actually have the vaccine anniversary on our calendar. It's like a day away from our wedding anniversary. So something that definitely has stuck with us for many years. But really excited about working with such an amazing team to get this done and to see it out.
That's great. Tell us, kind of maybe to start with, when you were developing the trial itself, you did this personalized cancer vaccine for patients who were high risk of recurrence, and maybe why you felt like that was the best place to start with. Was it mostly logistics? Was it kind of biologically, the rationale that you were looking for?
No, it's a great question. I think that the reality was mostly logistically driven. And in hindsight, I think there is some biology behind that. And so maybe mention what I mean. So at the time, this was a really homegrown operation. We weren't working with a company. So it was us that was shuttling the tumor tissue back and forth, getting it sequenced. We had a team doing the mutation calling, and we'd sit in a room. We had this what we call the epitope selection board, and we'd sit in the room for a few hours. This was initially prior to COVID, and then we switched to Zoom. But we would just go over every mutation, every antigen prediction, and sort of debate how to prioritize things. And then we'd obviously have to have it manufactured, sterilized, and all of these sorts of things. And so that process, it just took time. At the time, it probably took on average around 12 weeks. And so we were trying to think of what is the setting that sort of makes sense for this. At the time when we started, actually, this was before adjuvant Pembro was even approved. And so this just seemed like the perfect sort of clinical and logistics sort of setup. There was a clinical need because there wasn't an effective adjuvant therapy there. And we also had time. It wasn't, a rapidly progressive metastatic disease where we could take 12 weeks really to be able to make a vaccine and get it started. So that was kind of the thinking at the time. I think in hindsight, we perhaps got a little bit lucky with that timing as well, because certainly there's been lots of vaccine studies throughout the years, but including more recently, not in kidney, but neoantigen studies in a metastatic setting. And while this is not a formal comparison, it seems like the neoantigen vaccine studies that have had some success really do seem to be in this minimal disease setting. We've seen that in melanoma with the V940 Moderna-based mRNA vaccine. We've seen that out of Memorial Sloan Kettering with Vinod Balachandran's collaboration with BioNTech. Those successes have really been in the sort of after surgical resection, microscopic metastatic disease left over. That's where really those signals of activity for the vaccine has been. And I think it's been a little bit, honestly, just more of a challenge with sort of bulky macroscopic metastatic disease. And so as I sort of think about moving forward, it's not that I think, impossible that it would work, certainly in a metastatic space. But I think probably the prime place to start is that adjuvant setting.
Yeah. And when you kind of mentioned, that it took like 12 weeks and it was, a lot of kind of coming together, working as a team. Did you have a difficult time, recruiting and enrolling patients to be interested in the trial? And how was the logistics? Because, when I see 40 co-authors, I know a lot of it's the science, but what did it entail to kind of get this up and
I
going?
mean, huge steps along the way because there's a huge regulatory component of just getting, the INDs filed and processed, getting contracting to make peptides in a GMP sort of qualified facility with pharmacy, getting everything sterilized and organized. There's huge sort of components along the way. And then when the tumor is actually resected, who it goes to in terms of pathology, who's doing the second looks, who's going to be sort of the people on the ground doing the mutation calling calling and the antigen prediction. And then when we actually got together in a room, it was, people with clinical and biological knowledge, but there were peptide chemists who were there. There were people with, various expertise that they could all chime in on some of the logistics, things I've never thought about before in my life that, can this actually peptide that we really think would be a great target, maybe that just can't be synthesized because of technical reasons, or maybe it will precipitate in solution and we'll never be able to use it. And so those are all sort of things that factored in that made this a really amazing sort of team to work with. I think from the patient side, I'm just amazed by the patients that are enrolled in this because we really tried to learn as much as we possibly could. And so what was involved in the study from their perspective too was really a big ask. We did a lot of blood draws, a lot of large volume blood draws, including leukapheresis before and during the vaccine to really get a good number of immune cells to be able to analyze. We obviously had their tumor material, but we actually did skin biopsies along the way, both of the vaccine site to see what local immune reaction was causing, but also a sort of delayed type hypersensitivity analysis to really try to isolate vaccine reactive T cells. And so when you look what these patients were going through of so many injections and blood draws, they did it with a smile on their face. They were just incredible. They were, really excited about participating, which was just amazing to see. They were, the most important thing is how they're doing as an individual and their care. But there's so many of them that were just also excited about the program and the idea of contributing to it. And that's something that was unexpected, but I'm certainly grateful for.
Yeah, that's really amazing. I am constantly reminded about, it's really who we do it for, right? Wanting to understand the science and certainly move the field forward, but it's always only done because we have these amazing patients who are willing, to help us do that.